Speaker is Bernhard Horsthemke from the University of Duisburg-Essen, Germany. He will talk about epigenetics, innovations, and misconceptions. Can you hear us, Bernhard?
I can hear you. Can you hear me and see my presentation?
Yes, we see our presentation. Go ahead.
Okay, good morning, and thank you very much for inviting me to give a talk at this workshop. In 2012, Mr. Carey published a book called The Epigenetics Revolution, How Modern Biology is Rewriting Our Understanding of Genetics, Disease and Inheritance. And since that, many such papers, both in the scientific press and also in the lay press, have been published claiming that there has been an epigenetics revolution. I will discuss whether this claim is justified, and for this I will review the history of epigenetics and also the history of changing definitions of epigenetics, as well as the contribution of the major players in this field. And these are the people I will mention: Waddington, Nanny, Gurdon, Holly-Derricks, Kornbeck, Elvis, Jertel, and a few others. And in my talk I will highlight in yellow what I consider is the origin or type of innovation that these scientists has made. So it is mostly agreed that Conrad Hal Waddington, a British biologist, was the first to link development and genetics. And in 1942 he coined the term epigenetics and defined it as the branch of biology that studies the causal interactions between genes and their products which bring the phenotype into being. I think today this branch of biology is called developmental genetics, and one prominent figure in the field of developmental epigenetics is of course Eric Davidson, as we had yesterday. Now, the term epigenetics is comprised of two words, epigenesis and genetics. And epigenesis is attributed to Aristotle, so 2,000 years ago, though he didn't use the word, but he did observations in the developing of a chick in the egg. So epigenesis is the development of an organism through a series of new structures not preformed in the egg or sperm. The word epigenesis was first used by William Harvey in 1651. Aristotle did not use this word. I think this is an example, epigenetics, this is an example of the creation of a new scientific field by synthesis from two existing fields, in this case development and genetics. But bear in mind that this, at the time of Waddington in 1942, was a theoretical concept. This was 2 years before Oswald Avery found that DNA is the genetic material, and 10 years before its structure was elucidated. And in 1953 we saw the dawn of molecular genetics by the elucidation of a structure for DNA by Watson, Crick, and Franklin. Five years later, so we are already in the early molecular period, and in 1958, David Nanny introduced the term epigenetic control systems, and he published his work in PNAS, and you see the paper here. Now the interesting thing is, David Nanny did not work on development of multicellular organisms. He worked on tetrahymena, which is a unicellular eukaryote. And he is also famous for having established tetrahymena as a genetic model organism. The interesting thing about tetrahymena is that it has two cellular nuclei, a micronucleus, which is the germinal nucleus, and it is transcriptionally silent, and a macronucleus, which is derived from the micronucleus and transcriptionally active. Though at the time in 1958, David Nanny was not aware of the transcriptional activity of these two nuclei. This was shown only in 1969 by Borowski et al. Tetrahymena was crucial for the discovery of telomerase and RNA self-splicing, which gave 2 Nobel Prizes for the discoveries, and the histone acetyltransferase, which I will mention later. Nani himself worked on the non-Mendelian inheritance of cortical patterns and mating types in Tetrahymena, and probably by working on non-Mendelian inheritance, or apparently non-Mendelian inheritance, he started to think about development also in multicellular organism. And at first, in his paper in 1958, he refers to the Watson and Crick paper. He says, on the one hand, the maintenance of library of specificities, both expressed and unexpressed, is accomplished by a template replicating mechanism. And this is the genetic system. And this is the famous sentence in the Watson and Crick paper. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So it's a conservative, semi-conservative replication of the DNA. Now we come to the epigenetic control system. And Nani said, on the other hand, auxiliary mechanisms with different principles of operation are involved in first determining which specificities are to be expressed in any particular cell, and second, certain patterns of expression, although specifically induced, may be perpetuated in the absence of the inducing conditions. And this is what he called the epigenetic control system. The term epigenetic is chosen to emphasize the reliance of these systems on the genetic systems and to underscore their significance in developmental processes. And I think this is a novel concept within the existing framework of epigenetics. Let's have a look at the first part, determining which specificities are to be expressed in any particular cell. At the time of Nani, it was unclear whether the expression of specificities in any particular cell involved the loss of genes that are not needed, or the differential expression of genes that are needed. And it was 4 years later, in 1962, that John Gurdon showed by transplantation of somatic cell nuclei into enucleated oocytes that a terminally differentiated cell still contains all the genetic information needed to form all types of cells. Thus, cell differentiation is not based on loss of genetic material, apart from the immune system, as we heard yesterday, but on differential gene expression. These were certainly groundbreaking experiments, and as you know, Gurdon was awarded the Nobel Prize a couple of years ago. Still, the question was, how is differential gene expression achieved? And Ramon already mentioned yesterday that it was Jacot and Monod working on E. coli established the fact that the induction of protein synthesis is regulated at the gene expression level, and this regulation involves the binding of transcription factors to gene regulatory sequence elements. If E. coli needs to use lactose, it needs an enzyme called beta-galactosidase, which it doesn't need when it grows on glucose. And so the beta-galactosidase gene is shut off when glucose is available, but it needs to be shut on when lactose is the only carbon source. And this is achieved by the binding of a LAC repressor, so a transcription factor, to the operator or promoter of the structural genes, including the beta-galactosidase gene. So we have a transcription factor, in this case is a repressor, the LAC repressor, binding to here. The presence of lactose changes the configuration of the LAC repressor, it cannot bind, and so beta-galactosidase is expressed. So this was really a landmark finding, was also awarded a Nobel Prize, and this is also important for the development of multicellular organisms, which involve signaling cascade and gene regulatory networks. And of course, we have to see the work here by Eric Davidson. And he published this theory of gene regulation in higher cells in 1969. So let's come back to the Nanne's epigenetic control system, the second part. Certain patterns of expression, although specifically induced, may be perpetuated in the absence of inducing conditions. Of course, one way of perpetuation would be feedback loops, but this is difficult to assume that they will be perpetuated, these activities will be perpetuated through mitosis. And here comes the speculative hypothesis in 1975 of Riggs and Holliday and Poole. They independently of each other proposed that gene expression patterns can be affected and perpetuated by the enzymatic methylation of cytosine within CPG nucleotides. So this was really a speculative hypothesis, and it was based on bacterial restriction modification enzymes and the lac repressor model. This is certainly a groundbreaking idea, and again the interesting thing is that neither Holliday nor Riggs worked on the development of multicellular organisms. Holliday did research on genetic recombination and DNA repair in fungi, and Riggs worked on the lac repressor in E. coli. So what is the restriction modification system? It's the bacterial defense system. Many bacteria have a restriction enzyme which can degrade incoming phage DNA at specific sites, here shown by the HBA2 restriction enzyme, which cuts CCGG. And of course, the bacterium has to protect its own genome against the restriction enzyme, and it does so by methylating this recognition site so that the restriction enzyme cannot cut the bacterial genome. So DNA methyltransferases were quite well known when Riggs and Holliday made their proposal. The interesting thing is, in this proposal, Riggs and Holliday combined two apparently unrelated things, the restriction modification system and the lac repressor. And Riggs said the properties of restriction modification methylases are very similar to those of the lac repressor, which recognizes base sequences in duplex DNA. Methylations that occur in operator sites are very likely to affect regulatory protein binding. And combining these two unrelated things, I think, is a very good example of associated thinking, which is at the base of creativity. So let's have a look at the DNA methylation. So it was well known from bacteria already that there is a de novo DNA methyltransferase which can methylate the DNA on both strands. And this is most often associated with gene silencing because it often prevents the binding of transcription factors to the recognition site. The proof of DNA methylation's role in gene expression emerged much later in 1975. It emerged in the 1980s through the collective effort of several research groups. Now, very interestingly, both Holliday and Riggs also proposed the existence of a methyltransferase which has a preference for hemimethylated DNA, which is present immediately after the replication of the DNA. And this is a maintenance methylase transferase, and because it has a preference for hemimethylated DNA, it can read the methylation on the parental strand and then methylate the newly synthesized DNA strand. So this was proposed in 1975. The existence of this enzyme was proposed, and it was actually identified only in the 1980s by Tim Bester as DNMT1, and Tim Bester showed it to have a preference for hemimethylated DNA. Later, Holliday also considered what happens if DNA methylation is lost, maybe due to absence of the maintenance methyltransferase. And he wrote a very influential paper in 1987, again a hypothesis paper, The Inheritance of Epigenetic Defects. And he said loss of methylation will lead to heritable abnormalities in gene expression, and these may be important in oncogenesis and aging. I should say here with the asterisks, heritability was not part of Waddington's or Nani's concept of epigenetics, and this indeed is a problem of this hypothesis paper, but it was a very influential paper, and it was the paper which made me switch over from my early days in human genetics to epigenetics. So this paper brought me into the field of epigenetics almost 40 years ago. When Holliday made his proposal in 1987, There were only 42 papers having the word epigenetic in its title. In 88 it was just 43, and in 89, when I published my first paper on epigenetics, it was epigenetic changes may contribute to information in spontaneous regression of retinoblastoma, where I showed for the first time that a tumor suppressor gene can be methylated in cancer. At that time, the epigenetics community was a very small community. In 1989, my paper was one of only 62 papers with the topic epigenetic. In the early days, the areas of research were axon activation in females, genomic imprinting. Why? Because in these two situations we have monoallelic gene expression, although the two alleles are in the same nucleus, cell, organism, environment, and exposed to the same transcription factors. Still one gene is active and the other gene is inactive. And then we have normal versus malignant cells in cancer. So we then, of course, see very many papers on cancer. Until the end of the 80s, epigenetic was was equivalent to DNA methylation. Then it became known that not only DNA methylation, but also the nucleosomes and the histones plays a role. The DNA is wrapped around the histone octamer, the nucleosome, and the role of the nucleosomes as a regulator of transcription was first demonstrated by Cornell group in 1987. And you see here that most transcription factors cannot bind if the target sequence is on the nucleosome. It can only bind if the DNA is free of nucleosomes. So this was, I think, another groundbreaking discovery. Nucleosomes inhibit the initiation of transcription, but allow chain elongation with displacement of histones. And then we come to the histone modifications. Already in 1964, Alfred Mirsky suggested a role for histone acetylation and methylation in transcription. And in 1996, Ellis purified the first histone acetyltransferase from tetrahymena micronuclei. Remember that Nane had established tetrahymena as a genetic model organism, and Ellis showed it to be a transcriptional coactivator. This was certainly a groundbreaking idea and discovery. And here's the paper, tetrahymena histone acetyltransferase, a homolog to yeast, linking histone acetylation until gene acetylation. Now we know very many histone modifications, and I don't have time to go into these. Some of these histone modifications are associated with active promoters or active enhancers, others with repressed promoters. So there's a huge variety of different modifications. Until today, we know more than 100 chromatin modifying enzymes, both DNA and mainly histones. The important thing is that chromatin modifying enzymes cannot read DNA sequence contact and are recruited to specific sites by transcription factors. They are partners of transcription factors. Most of the modifications do not survive DNA replication and thus cannot perpetuate cell-fate decision. There are only 3 exceptions, which are the methylation of DNA, as I mentioned before, by the presence of the DNA methyltransferase 1, which recognizes helimethylated DNA to methylate the newly synthesized DNA strand. And we have a polycomb repressive complex 2, which can read. So I should mention that during DNA replication, the old histone octamers are recycled, but the cell needs twice as many histone octamers because we now have 2 DNA strands. And newly histone octamers are incorporated, which lack histone modifications. And this PLC2, for example, can read the methylation on histone 3 lysine 27, and then methylate the same lysine side chain on the newly recruited histone here. A similar thing occurs with a different protein for methylation of lysine 9 at histone 3. In 1996, Rix et al. Defined epigenetic as the study of mitopically and or meiotically heritable changes in gene function that are not caused by a change in DNA sequence. This is a widespread definition, but I don't like it. because this is a negative and ambiguous definition. Many researchers include RNA modifications, microRNAs, prions, and lots of other things, which I think makes little sense, and blurs a distinction between perpetuation of gene activity and heritability. This is probably partly due to the unfortunate use of the word heritability by Holliday in his 1987 papers. Because, please remember, most of the chromatin modifications do not survive mitosis, let alone meiosis, as I've mentioned before. And interestingly, many researchers who work on histromodifications prefer the Rick's definition, although their modification is barely mitotically or meiotically stable. A less ambiguous and more pragmatic definition was by Adrian Bird in 2007. Chromatin modification served to register, signal, or perpetuate altered activity states, which basically includes only DNA methylation, nucleosome space, and histomodification. And I think this is the better definition. But as you will see, or as you see, epigenetics is probably the only scientific field where the researchers can't agree on a uniform definition. So today there is no universally accepted definition of epigenetics. The field overlaps with transcription regulation and chromatin biology, and this fuzziness may have contributed to the explosion of epigenetic papers in the past 30 years. And here you see the explosion. Again, a search with the term epigenetic reveals that there is an explosion starting here. Now there are more than 160,000 papers, and 50% of those papers are on cancer. Most often it's DNA methylation serving as a biomarker in cancer. Now I come back to Mr. Carey's book, The Epigenetics Revolution, how modern biology is rewriting our understanding of genetics, disease, and inheritance. Basically, this book, and as I mentioned, many other articles in the field, scientific field and in lay press, they make two claims. The first claim is that diet, stress, and trauma affects the epigenome. In short, you can control your genes. And the second claim is these changes can be transmitted to offspring. In short, you are what your parents and grandparents ate. Now, interestingly, the proponents of this epigenetics revolution, they don't realize that these two claims actually contradict each other. Because why should we be able to control our genes through chromatin modifications, but not be able to change this chromatin modification that we have inherited from our parents? So let's have a look at the first claim. Diet, stress, and trauma affect the epigenome. As many people say, we can control our genes. Here's a figure from Wikipedia, but you see similar figures all over the world. And it says methyl group, an epigenetic factor found in some dietary sources, can tag DNA and activate or repress genes. So there's a shortcut from the environment, the diet, with methylation straight onto the DNA. Which, of course, I think is nonsense. What we have here, of course, the environment has an effect on gene expression, but it is through activation of signaling pathways, activation of transcription factors, cellular differentiation, proliferation, cellular composition of tissues and organs, and hormonal and metabolic set points. So yes, there are DNA methylation patterns they can change, but these reflect these changes rather than cause these changes, so they reflect these changes, and they can serve as biomarkers, and because DNA methylation is rather stable, they are excellent biomarkers. So where comes all this from? And this is connected, I think, to the paper by Waterland and Randy Jertel in 2003, Transposable Elements, Targets for Early Nutritional Effects on epigenetic gene regulation. He studied agouti by the yellow mice. The agouti gene is expressed only in hair follicle, but in this mutant mice there's a retransporter in front of the agouti gene, which can lead to ectopic expression. So this interstitial A particle promoter can serve as a ubiquitous promoter. And interestingly, offspring of mothers carrying heterozygous animals have a range of phenotypes, from yellow antobese to agouti or normal rate, and everything in between, from slightly mottled to obese to heavily mottled. The authors say that maternal methyl donor supplementation, so they supplemented the nutrition which they gave to these pregnant mice. Maternal methyl donor supplementation with folic acid, vitamin B12 is associated with a shift in phenotype distribution to more agouti and to more IAP methylation, so a shift to more of this phenotype. And they conclude that dietary methyl donor supplementation alters AVI offspring phenotype by increasing CPD methylation at the AVI locus. This paper has been referenced in many other articles, and it has made it into the lay press, but the conclusion is not justified. The authors confuse correlation with causation. And several papers appeared later, in 2010, here saying that CPD methylation of a silent-controlling element in the murine AVI allele, this is the AIP element, is incomplete and unresponsive to methyl donor supplementation. And another paper in 2021, variably methylated transposons are refractory to a range of environmental perturbation. And there are a few other papers rejecting the Waterland and Jertel paper, but Jertel still goes around telling his story. And these papers, which demonstrate that this was flawed research, have not made it into the lay press. So many people in the lay audience still believe that nutrition can directly change methylation of their genes. Now I come to the second claim. These changes can be transmitted to offspring. You are what your parents and grandparents ate. First of all, transgeneration epigenetic inheritance. Yes, it does occur in some plants. RNA-based inheritance occurs in nematode. It has been labeled epigenetic, but I think it's not epigenetic inheritance. It is RNA-based inheritance, and it needs RNA-dependent RNA polymerase, which mammals do not have. There is no convincing evidence for transgenerational epigenetic inheritance in mammals. Studies claiming to show this suffer from methodological issues, statistical issues, p-hacking, arcing. Arcing is hypothesis after results are known. P-hacking means you use different p-tests and use the one which gives the smallest p-value, and lack of independent confirmation. Here's one example. It's the famous Överdick-Skaldick study in 2002. It's a small region in the northern part of Sweden where food records and harvest records and food prices were available, and this authors published that cardiovascular and diabetes mortality determined by nutrition during parents' and grandparents' low growth period. They say that boys, enjoying food available had grandsons, at this time, that boys enjoying food available had grandsons which lived on average 6 years shorter than the grandsons of starved boys. This was not observed in granddaughters, was not observed when it was through the female germ line, but also this was very well covered in the lay press. But what was not very well covered was the Uppsala birth cohort in 2018, where Gerard van Berg published, we were unable to reproduce previous results for diabetes and cardiovascular mortality. And this was an even bigger study than the Överdick-Skaldick study. There are two, also in mammals, there are two barriers to transgeneration epigenetic inheritance. There's the Weismann barrier, saying that information in somatic cells cannot flow into the germ cells. And then there is erasure of DNA methylation in primordial germs in the early embryo. So DNA methylation patterns will find it very hard to survive primordial germ cell development and early development. So I'm coming to the end of my talk. Molecular epigenetics is a rapidly expanding field, but I do not think that it rewrites our understanding of genetics, disease, and inheritance. Reasons for the widespread misconception of epigenetics, to my impression, are first, a confusion between correlation and causation. Second, the ignorance of established facts, for example, that transcription factors are determining cell fates and chromatin modifying enzymes are partners, but they don't do it on themselves. And ideology, many people emphasize nurture over nature. there's quite many people who don't like the idea that part of our behavior and how we look like is determined by genes, so I say it's our nurture. And now I think they have a scientific reason for their impression that nurture determines our nature. And then of course there are scientists, journal editors and lay people who like paradigm shifts. So this is coming to my conclusions. So I've mentioned all these scientists here. When we look at the original type of innovation, we have a theoretical concept synthesis from two existing fields, novel concept, groundbreaking experiments, but then we have Holliday-Riggs, groundbreaking idea with DNA methylation, Cornbeck, groundbreaking discovery. Orfrey and Mirsky had already groundbreaking idea with regard to histone modifications, and Ellis by discovering the role of histone estrogen transferase. And when Jörtl and others, which I think is a misconception, mistakenly regarding as epigenetics revolution. And my take-home message is most of the innovations establishing the field of epigenetics were based on groundbreaking ideas which extend previous knowledge, and rather than groundbreaking discoveries. I think that associative thinking played an important role. Remember how Holliday and Riggs put together the restriction modification system in E. coli and the luck repressor model. But associative thinking needs knowledge and memory, and we heard already yesterday that our students nowadays may have less of knowledge and memory. And I think epigenetics has not displaced previous concepts. The so-called epigenetics revolution is a misconception. So I thank you for your attention, and I will stop my presentation. Okay.
Thank you, Bernhard. Great talk. Any questions? Yes.
I have a simple question. Maybe it's too simple. I'm a physicist and have no ideas about genetics and so on, fundamental. So, but it came the question.
If there is a question, I don't understand it.
What that? Is that
Here, come.
so? I'm a physicist and not genetic, and I have a simple question, maybe too simple. Is there any relation between the development of epigenetics and the ideas of Lysenko? Of course,
Yes, of course. It's a very hot topic because there are some people in the community who try to revive Lamarck. Somehow it's very attractive, but so far I'm not convinced that there is evidence that there is inheritance of acquired traits. You may have, Waddington, for example, worked on genetic assimilation. He could show that exposure of Drosophila to some environmental conditions and selection for phenotype can change the genotype, but the explanation for genetic assimilation is that there are genetic variants already present in the Drosophila, and later when you have selection, these variants later take over, but it is not the inheritance of acquired traits.
Any more questions? Okay, let's take Bernhard again.